My name is Immaculata DeVivo, known as Mac, and I'm a professor of medicine at the Harvard Medical School. I also am a professor at the School of Public Health, and my research focus is cancer mechanisms, specifically elucidating the mechanism with respect to etiology. And I work with the endometrial cancer as my focus, and over the years we have acquired enough information on risk factor data as well as genetics, but the risk factor data is overwhelmingly more established, and enough of it to aggregate to make predictions with respect to disease risk in the near future.
Got it. And just to clarify for now, so risk factor data, what kind of data are we talking about?
We call it environmental data, but it's not about climate change. You know, the environmental data, lifestyle data, anthropometric data, hormone use, parity, reproductive factors, we call that risk factor data.
Got it. Okay, come back to that later.
Sure.
And Dr. Kraft.
Yeah. So I'm Peter Kraft. I'm a professor of epidemiology and biostatistics at the School of Public Health, Harvard School of Public Health. My research largely focuses on the genetic epidemiology of complex traits. So complex traits are things that are influenced by many factors, including genetics and risk factors, environmental risk factors. So a lot of my research is focused on gene-environment interactions. And I came to epidemiology by way of statistics, where I came by way of math.
Got it. And you came by way of?
The lab.
The lab.
I came by way of studying cancer at the bench, as we call it, and then in humans, actually, when I was at Columbia, and then moved to the West Coast, Stanford, with a slight detour through Berkeley. But at Stanford, I really, have to say, earned my lab chops by working on mouse models of leukemia.
Right. So in my world, this is like observers and theorists, and like people who deal with data they acquired themselves, and people who deal with no data in astronomy, or data that other people acquired. And so, to be fair, we have sort of a little bit more of a kind of go get the data at the lab to start out with, and figure out statistically, mathematically, how to model the data and how to understand the data.
Ish,
fair?
yeah. So
Ish,
I mean, I'm
yeah.
also involved with collaborators. I'm not the one writing the questionnaires or mailing out the studies, but I'm working with people when they design the study.
Right, but what's important for us for later when we talk about this more is that there's kind of a spectrum of people involved.
Yes, absolutely.
Absolutely. And one of the reasons I switched over to humans when I came to Harvard was that, and I paraphrase a very famous comedian who said, "We've already cured cancer in mice. Let's focus on humans."
Who said that?
I want to say Jay Leno, but I could be wrong.
Okay.
My memory's not as great as it once was with that.
Somebody else is studying that. Yeah.
Someone else is studying that. But I think if it's critical to the discussion why I went from this hardcore mouse model person to landing into the Harvard cohorts, which have these population-based studies. The idea that you can track people over many years, up to 40 years, I believe now, 45, and then integrate the lab data, to me was just the most holistic way to sort of answer these questions related to cancer.
So now that I know a little bit more about each of you than I knew before, I had some questions and some information kind of about like what we're doing these days and what we're looking for. And so the whole prediction project, as you probably know, is about kind of tracing the history of prediction from ancient Mesopotamian sheep entrails, which we love talking about, to the kind of stuff that you both do. So modern simulation and prediction work. So actually the first question I want to ask to kind of get the story completely straight is, when I say prediction, I might not mean the same thing that you mean when you say prediction. So I've heard Mac use the phrase prediction model a lot, and I've heard other people in epidemiology say that. And I use the word simulation kind of interchangeably with model sometimes, but then people in the broader world thinks sometimes I'm talking about 3D printing and models, which is clearly not what any of us are talking about, okay? But when I say prediction or model, I know what I mean, but can you each explain what you mean? Like, what's a prediction model? What's something that would be done in epidemiology that would not be a prediction? Like, what's the difference between a prediction and not a prediction?
Well, broadly speaking, when I mean prediction with respect to risk factors is in aggregate. We have a handful of, I'll give you a real life example.
Yeah.
So we have a handful of risk factors that are really well established to be well established and associated, don't really align, but they're established to be related to endometrial cancer. And so we know that each one of them imparts a particular risk assets, a particular effect. So the idea is that if you could put them all together, and I'll have Pete speak to the math, you can get a better picture as to a woman's complete risk, given the risk factors.
And so just to be specific, like give a few risk factors.
Sure. So the best risk factor for endometrial cancer, again, and I'm not going to go into the weeds of subtype, is BMI. Obesity is a main risk factor for endometrial cancer, but also parity, the number of children or lack of children, and hormone use. Another very large risk factor for endometrial cancer.
So the last two of those are not genetic or not obviously genetic.
Right.
BMI could have some genetic component. And so one of the questions I want to ask you, Pete, is how do you then, when you try to make a prediction model out of this kind of data, how do you disentangle? Like, you want to look at the joint story, but you also want to understand what's at play, right?
Yeah. No, that's an excellent question.
Yeah.
So I think it's when it comes to understanding the joint story, maybe the biggest challenges up front is getting the data on all the relevant risk factors. Because a lot of times we'll have a study where we've invested in getting genetics on everybody, but we don't have the detailed information about their diet or circulating plasma levels of this, that, or the other, right?
Right.
And then the other studies will have the other things, but not the genetics. So just getting that all together at once is a big part of the battle. And then once you have it, you have some flexibility in terms of how you model it. Basically, you can include it all in the model simultaneously. And it becomes, on some level, you can even do sort of black box prediction modeling.
Yes,
if that's what you're interested
I
in.
definitely want to talk about that
right.
black box idea, because one of the things that we talk about a lot in the modern predictive systems is artificial intelligence and machine learning and things where, and in this very room, we've had conversations with Ben Schneiderman and with other people about kind of whether or not you need a hypothesis in science. And so Mac knows that a few weeks ago we were in Italy and we were talking about this, and I was talking about it with a bunch of medical or health-related people. And I had to try to make an analogy between physics and medical research. And that was the first time I ever did that. And so part of what's in this little stack of paper here that I want to show you is that, and I want to try it out on you. But something interesting that happened was this. And so we'll put this picture into the video if we use this part of the conversation. But since Mac was there and you weren't, you can take a look at that picture.
Okay.
And it's a picture of a very foggy day in the morning in Venice on the Grand Canal, and you're going down the Grand Canal on a public boat, a Vaporetto. And you've been down the Grand Canal before, and you know that there's some buildings that look kind of like these buildings that are past there, but you don't know, is there a shark? You've never seen a shark before in the middle of the Grand Canal, but because it's so foggy, you can't see it, right? And so one thing that we've realized is that people are used to being able to see kind of far away in any direction in space, okay? But in time, they're not used to that. And so there's this idea that prediction is wanting to know what's already there in your future, but you just can't see it, right? Is there a shark waiting for you? And so there's this idea that some people have, right, that I don't know that I necessarily subscribe to, because especially with this interaction with environmental features, but I think a lot of people get the idea that they have some future that's written in their genetic code, and that if only you people could understand it, they would know their future. And yeah, I could bring up things like 23andMe or those other things you don't necessarily like. But go ahead and tell me what your opinions are about how much of it is that it's all there and we just don't know how to interpret it, and how much of it is interaction with environmental factors, and how much of it is we just don't know.
You're hinting at predisposition.
Yeah.
So, and that's actually, how Pete and I, I'll speak for myself, we make our living working with the kind of genetics that is non-Mendelian. Okay, so it's it doesn't quite give you your fate just based on your genes. So we're talking about susceptibility genes that a rough definition would mean that they have to work in concert with other genes, or I'm not going to go into the weeds of genetic loci, or with some environmental factor to give you the effect. So, I mean, the studies, the data that we're all involved with suggest there are a number of predisposing genetic markers, okay? And a lot of them, or many of them, I don't have any, I'm not going to enumerate for you, but some of the studies suggest that when they work in concert with some environmental exposure, the effect is profound. Not profound, but you see the effect.
Right.
And I wish I had a great example for you right now, because I can't think of one, but I'll give Pete the time to think of an example where you have some sort of genetic predisposition, but you don't actually see it until you have the...
What about something like, is skin cancer that way? Like if you don't spend any time in the sun, maybe you're not going to have a problem, but if you do, you do?
Well, skin cancer is not just one cancer. Like I said, there's a melanoma, which is the most aggressive, and there's squamous and basal. And melanoma actually is the one cancer that doesn't really, the sun exposure isn't as established as like squamous and basal. And so it's, I'm not trying to be like cagey. It's a very interesting question, and it's a question that, Pete and myself are, dedicating our research on to understand this interaction where you have an inborn or genetic predisposition,
Right.
and why is it that, Mary has this genetic predisposition, she doesn't get cancer X, but Joe has it, and he gets the cancer X.
Right.
Is it because Joe is a smoker? Is it because Joe is heavy? That's what we're trying to tease out. And one of the advantages in a long-standing longitudinal study where you have complete data, or as complete as possible, is the opportunity to sort of address these kinds of questions.
Because you have a lot of examples, a lot of cases.
We are fortunate to work within, like I said, the Nurses' Health Study, where we have updated data every 2 years on a lot of women, as opposed to many studies, just because of the way they were designed or the way they ended up, don't have updated. They have maybe 1 or 2 data points on an individual.
This is the value of having lots of data and longitudinal data.
Longitudinal with repeated, we call them measures, or updated data on an individual.
Right. Well, let me ask you. So Pete, I want to know what you think too. But let me just add a little twist to it. So when we talk about the ancient divination systems and the people who were like consulting with the king and saying whether or not to go to battle or whatever, if they tried to, I'm not saying she's hedging, she's being honest, okay? But if they tried to say, "Whoa, I don't know, it might be like this and it might be like that," whatever, they could get killed. Right? So they had to seem that they were more sure than they were, although there's some debate in history that maybe they didn't always have to do that, but there's certainly an argument to be made that they did. And so another question that I wanted to ask both of you that we can think about now in this context is about uncertainty, and about how you convey the uncertainty to each other as scientists, and how you convey it to the public, and how you convey it to doctors who are going to make decisions based on these things.
Right.
So
that was exactly what I was going to try and address.
Tell
I think
us,
that's a really important question, and it's something we're all struggling with right now.
So you don't want to be killed by the king.
No. Or, if you're consulting with a patient, you want to give them honest advice so that they can make informed decisions.
Right.
So I guess there's sort of uncertainty on 2 levels. The first is I certainly don't conceive of the future as something that's sort of there, written in stone, and will happen, and we just have to sort of look and see what it's going to be.
Good.
There's all kinds of possibilities.
I shouldn't say good. That was my personal opinion, but good.
But even if you think, like, if we knew the position of every molecule in the universe precisely, and then we would know exactly what's going to happen, we're nowhere near that, right? So there's tons of uncertainty. So, in the risk models that we build, for example, for like breast cancer, it's your 5-year risk of getting breast cancer, given that you're 65 years old now, and I know this, and the other about you. It's 6%, right? So out of 100 people who look just like you, 6 of them are going to get breast cancer. So there's that. It's not deterministic. It's like we can't say you will or you won't.
Right.
It's this percentage. The other part of the uncertainty is that number is coming from a model that we fit that itself has some fuzz around it, right? That's our best estimate, but maybe in our data set and using our models, that's actually 6% plus or minus. It could be a little higher, it could be a little lower. But if you looked at a different data set using a different model, you would get an estimate of 8% plus or minus a little bit. So, and that's conveying that uncertainty is the extra challenge that I think we're wrestling with. So the climate people, the weather people have sort of figured this out when you look at the hurricane maps, right? You've got like 5 different models,
people, yes,
right?
the climate people know.
Sorry,
Yes.
good point. No,
Yes.
Just the like short term, like out a week ahead.
Yes.
You can sort of see the models start to converge.
Right.
But at the beginning, you've got 3 or 4 different
Even so,
predictions.
it's so funny that you use that example because, I teach a freshman seminar in conjunction with this, and I always use that example about the hurricanes and the path of the hurricane, spreading out over time. And it's not that the hurricane's going to get bigger, it's that they don't know exactly where the center of the hurricane is going to be from all different models. Even Harvard undergraduates, when they look at those things on the internet or on TV or whatever, they don't completely understand what they mean. They mean like, there might be they see these like contours of uncertainty, and you think, as scientists, we're like, wow, that's beautiful, that's a great example. The weather people have it all worked out. Even Harvard undergraduates still don't completely understand. I mean, they do by the end of our seminar. But anyway, but for climate, the entire motivation, and I probably told you this a few years ago when we first talked about this, but the entire motivation for the prediction project is so that when people hear uncertainty associated with climate change, that they know that that's a measure of certainty, not a we have no idea.
Right.
And you face exactly the same problem, because if you start being as honest as you are with me, with people who aren't going to appreciate the honesty, not because they're stupid or ignorant, but just because they want an answer, right? Then it's not always good, right? So you have to decide, and it's very dangerous. And one of the hardest things about this effort, this whole project, is explaining that. Explaining that uncertainty is a quantification of certainty.
And it's not a bad thing.
No, and it's
a thoughtful way to actually do, I don't even want to call it science, but just
and
information.
So then you can talk about like modeling uncertainty as itself a field, right? And then this is going to get us back to artificial intelligence, because once upon a time, when we learned how to do least squares fits in science, there was a way to calculate, based on the errors of every point, what the uncertainty was in the line, right? And that's a very sort of formulaic way to do things. But we all know that that's pretty much not the way most statistics work now, and that there's a hard job of, especially with very empirical work, of figuring out how to correctly estimate the uncertainty. So that's a problem that's sort of super subtle, okay? But here's where I want to ask you about this. So when I get to the whole artificial intelligence, right? So Max saw this story in Italy, but I'll put it here, and we'll put it on the screen if this part of the conversation winds up being interesting. So, all right, this is a spectrum here from noticing a phenomenon. We'll talk about Isaac Newton here in a minute, but noticing a phenomenon to purposefully observing it, to collecting data about it, to making a rule that explains it, to possibly making a real theory that means you understand why that rule would work, and then maybe you just want a rule or a theory to explain something that's going on, or maybe you actually want to be able to make a prediction. And what's interesting is that if you look at the history of science, and you look at, say, the ancient Babylonians, and by the way, these people are part of a broader project that's part of the course called The Path to Newton, and I didn't bring that because it's 7 feet long, and we can talk about that another time, but Mac's going to make The Path to Darwin, and you can help. But anyway, so...
She's optimistic.
Yeah, I'm very optimistic. You're going to do it. So if you look at, say, our friends, the ancient Babylonians, represented here by my friend Irving Finkel, who's the world's expert on their Mesopotamian tablets, they did this, okay? So they noticed phenomena like eclipses and other kinds of things in nature. They definitely observed them extensively. They definitely recorded huge amounts of data. They made some rules and occasionally predictions, but not really based on this. They made predictions based on empirical data of like eclipses and things like that, they should actually get a prediction box here, okay? Aristotle, not good, okay? So Aristotle noticed stuff, maybe kind of sometimes made some observations, skipped over data and rules, and just started making theories that he thought explained things and that made predictions. Now this is the interesting one, okay? So Kepler has these laws that describe the positions of planets quite accurately and correctly if you understand what gravity was or is. But he doesn't understand. He didn't know anything about gravity. He just knew that if you take orbits and instead of having circles, you make ellipses and you put the sun at one focus, this comes out really well, okay? So he goes, phenomenon, observation, data, this is other people's work, but he has all this, and he makes a rule, which is called Kepler's laws, and he can make predictions, but he can't make any explanations, okay? So Newton, the thing that makes Newton's theory of gravity the only, up to this point, really purely predictive theory is that you have this whole story and you can make a prediction where you have no data before. I can drop the pen and I still know the answer even though I never dropped the pen before, right? So then I told you I had to compare physics and biology, okay? So here's Kepler and Newton, and up here we have Mendel and Darwin. And this is part of the crux of what we were talking about before, where Mendel, for Mendelian traits that Mac was mentioning, you get to this rule with which you can make pretty solid predictions, right? But Darwin here, not really a rule. There's a theory. It gives good explanations of stuff that happens, but it doesn't make fabulous predictions, right? So let's say you want to get past there. Then you can do this, okay? And so this page says the future of the future. And so if this was the sort of 20th century gold standard, Newton's theory of gravity, let's take Google Maps as the 21st century gold standard. And then we go phenomenon, observation, data. Nope, don't need a rule, don't need a theory, don't need an explanation. I can just make a prediction based on a lot of data. So I'm guessing you can see where this question is going, right? Do we want to do this in the world of health and genomics? Like, do we care about all this stuff? And we have one person, Joel Dudley, who I think he cares a little, but he's perfectly happy to go from here to here. And the question is, should we, and should we sometimes?
So I think this is a really nice way of framing it, and I think the key is, what do you want to do with your prediction? So one could just be, I want to identify people who are at high risk of breast cancer because I'm going to screen them earlier.
Okay.
Earlier and more often. So the intervention there sort of has nothing to do with the underlying mechanism. Like, it's going to work. Like, we're going to catch the cancer early.
Right.
The other would be something like, oh, you know what? I've noticed that C-reactive protein. If you have high C-reactive protein levels, you're at a high risk of heart disease. So I'm going to give you a drug that lowers your C-reactive protein. There's actually no guarantee that that's going to lower your risk of heart disease, right? It's a great predictor. Like, if you have a high CRP level, chances are you're at high risk, but it doesn't mean that by intervening on that particular factor and reducing it, it's going to change it, right? So if you're looking for sort of a causal explanation, this sort of goes more to the theory part, right? This is why theory is important.
Right.
Like, by intervening on this factor, we can make a change.
Right.
That's not necessarily something that's going to come out of the black box prediction.
Right, and so if you so at some level, it's like if you want to just make an effective intervention, then skipping from here to here in a big cohort where you know a lot, and then seeing some set of markers for somebody that's like, ooh, that's really definitely do something, that's good. But if you want to actually understand, if you want to get ideas for future work and future interventions, you have to kind of understand the mechanism a little bit more, and that approach will.
It's also the idea behind medical doctors, and I say this in the best way. So if you are you go into the ER and you have, I don't know, incredible kidney pain or something. You don't know where that pain is coming from. Is it a stone? Is it some sort of genetic abnormality, or physiological? You have no clue, and the doctor shouldn't know where that origin is. He needs to fix you right then and there to get you out of your pain.
Right.
So I don't know where that would be on this continuum, but he would give you an IV of antibiotics.
Well,
or,
actually, no, that's a really good example because there are these systems now, right, where they're talking about putting intelligent agents in hospitals that would do the kind of thing we were talking about a minute ago, where they take that patient's symptoms and then they put it through some magic algorithm that says, ooh, that person might have, et cetera. Or you rely on just the doctor's knowledge to go, oh, I've seen this before and I have a theory that da 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 da. And, you know, I think it's some interaction of both.
But my point in telling that is that the main distinction I see between someone like me who's interested, oh, well, that's interesting. Why is that? You know,
Right.
what
why is
yeah,
that?
why, right?
Right.
The how. Whereas the doctor would
doctor
his
just
job
wants to
is
answer.
to, oh my God, I have to treat this person, give them, I don't know what they would give, because again, as I say, I'm not a real doctor.
Right.
Because that's the job. That is in the job description of this
What
person.
has been your experience? So, I mean, both of you, you're at the medical school, School of Public Health,
The same-
medical
the
school.
same quad,
that
right?
world, there are people in white coats, you know, walk around
Sure.
and treat people. And so when you actually talk with those people, what is their reaction? Do they want to understand, or do they want a magic system that tells them what to do?
Oh, that's a great question, because you can imagine
That
you
there's
will have the
all
spectrum.
types. Yes, of course. But
You will have the spectrum. I mean, I work with collaborators who will go unnamed who bring me blood samples and mice samples, and they have patients because they're oncologists, but they want to do the deep dive. They want to understand, and I would have to believe that they're also treating the patient on the other end.
Right.
So in my experience, it's run the spectrum from, I don't really care what the cause is, I need to stop the bleeding, to, gee, what really is the cause of that bleeding? So I don't know about you, Pete.
Yeah, I mean, I think this is, it's a cause of much discussion and debate right now, and there's definitely a diversity of views. So even setting aside, like, we understand, like, you can put genetic information in the medical chart, right? Do docs want that? Will they use it? Do they understand it? Do the patients understand it? How do we communicate it effectively? Does it actually change medical care? These are all, frankly, open empirical questions, and you're going to get people, doctors, who will be champions, who think that this is the way to the future. Yes, give me my Apple Watch, give me my 23andMe. And there will be others that you know what, I'm not convinced that this is giving us anything extra, and it's just going to be a major headache, and there'll be all kinds of people coming in with false positive results, and we're going to have to...
Well, actually, we had an interesting reaction. I mean, I may be sort of overstating it. We'll have to go back and look at the video. But Megan Murray, we interviewed her about, field epidemiology, and we also talked with Carolyn Buckey, and I understand that they're pals, and but it's clear that Megan was more at the going on instinct, and I've done this before, end of epidemiology, rather than give me the big data. And I think that there are crossovers where a technique, I've seen this in my own work in computing, is really crappy, and it's only sort of the early adopters who will use it at first, and then it gets to the point where it's kind of amazing and everybody uses it. And photo recognition is an example of that now, right? If I go on my phone and I ask for pictures of a person right now, they'll be 99.9% correct. But 10 years ago, not true.
But that, actually, the photo example is a good one, something that I want to bring up in terms of going from data to prediction, just sort of throw it in an algorithm.
Right.
How did Google or whoever,
Yeah.
how did these artificial intelligence folks learn to be able to say, oh, that's this person.
Right.
Look, oh, that's a picture of a flower.
Right.
This
Lots
is a
of
picture
training.
of a car. Yes.
Right.
Yes.
Right. You need a big training data set, right?
Right.
And it's one of the challenges facing people in facial recognition right now is that their algorithms are not as good at people who are not white men.
That's right.
And a large part of that is because of the training data.
Exactly.
The training data are all white men. Same thing for medicine. It's a huge challenge that a lot of the training data now are white men.
Right.
And there's I meant to look this up before I came in, but there's a book that came out recently about how the world is designed for men and how that is affecting women's health, right? Like if the seatbelt is if safety is designed around an average man, and the average woman doesn't fit that body type, then no wonder they're getting more injured in accidents.
So and you're interested in this too. I know that you're trying to get more and more data for diverse populations.
Diverse populations, that's correct. Yeah. But going back to a little bit of this continuum here, some of the docs that I work with, the medical doctors, they're sort of pressured to learn a lot of this genetic prediction, et cetera, because of the precision medicine. And I know you want to go there, but the precision medicine, no, I'm not upset by it. I just think that there's this push on to profile the patient with such accuracy, which is ridiculous because there isn't any, as we've been saying.
But there could be in the future is the question. There isn't now.
At this, I would have to say yes at this moment. But this
yes,
precision,
that there isn't.
There
that
isn't.
Kind of accuracy.
But the precision medicine, which is guiding, guiding a lot of the research, as you know, and we know.
Just for the viewing public here, by precision medicine, we mean having actual genetic information about a person that could customize medical treatments to that person.
Yeah, it's tailored. It's not one size fits all. It's a tailored approach to the individual.
And if that would work, great. We're talking about whether or not it works now, right?
Right.
And could it ever work?
Yes, it could. I mean, yes, everything is, right.
Actually, the shtick with precision medicine is usually it's the right drug for the right patient at the right time.
Right.
Yeah. So the physicians that at least I interact with, this is the drive to know more. The deep dive is really it's predicated on this precision medicine idea that.
Well, do you think that there's a sensible way to ramp that up slowly? Like right now, if there's a drug where they have all kinds of data about how well that drug works in just people of different ages, nothing complicated, and then the person prescribing the drug knew that. They'd probably take that information into account. So is there anything that's that simple that's
for
working?
Coumadin or warfarin, there's some, I don't know, there's some people who actually do screen. So warfarin or Coumadin is sort of a drug given for clotting. And so the old days is that take this and, if you don't die overnight, that was the good dose for you, right? So there is, and I don't know what it is, but the crew can maybe look it up and we can insert it. There is a way you can screen to determine a particular drug to know if you can sensitive to it or not.
I mean, there's been some real success stories in oncology.
In oncology,
right? So where you have a tumor that has a particular mutation. And there's a drug that works fabulously well, but only if you have that mutation. So if you get that to a random patient, it's not necessarily going to work. But for this subset of people, it works really, really well.
And so it's sort of starting to work, which gives people hope that it could work better in the future. And so then the problem comes in, it's sort of like the first round of artificial intelligence 40 or 50 years ago, and people went, that's never going to work. And then people, sort of changed their approach. But there was this time where AI, like as a term, was... you didn't say that in computer science because that had failed. And so what I'm worried about is you have the 23andMe's of the world where people think they're going to learn everything about their future now, and then when people see that, this doesn't actually work so well, that they're going to give up on it as a predictive system for the future.
So actually, to go back to the oncology example for a second, like the places where it works, those drugs were really developed on the basis of biological knowledge. Like we think this is how this is how the drug is working, and people who have this particular mutation, this is why the drug will work in them and not others.
Okay.
The excitement now is, can we sort of just take what we think we know about what the drugs are doing and what we think we know what these mutations are doing, and sort of machine learn our way to finding a treatment for a particular person.
Right.
So that's what we don't know how that's going to work yet.
Right, and Joel Dudley, who will put his interview from the Undiscovered in PredictionX, he's very optimistic about things like that. And there's a whole crowd of people who are optimistic about things like that. And there's this story about Sage BioNetworks, and Joel was involved with them, and that they had all this data from drug trials that were not needed by the drug companies anymore, and that just with giant statistical studies managed to find drugs to cure orphan diseases, and that they keep doing that. And assuming that's correct, in some cases, their claims are that it's due to kind of what you were saying about the nurses study, that's just this tremendous volume of data, and that it's enough of a training set to find these, otherwise very weak hidden trends. So is that valid?
Yeah, I mean, for me, like I'm a statistician, so I'm going to say, yeah, you give me a big data set, and we'll learn something interesting.
Okay, right.
But I mean, big is big in this case because a lot of these, for patients, when we find a mutation, it's 1 in 1,000 or 1 in 10,000, and then you need enough of those people to be able to understand what works, and then it's all observational, right? So these aren't actually, for the most part, these aren't trials. It's sort of this person got this drug. Was there something else, some other reason why they got that drug, and that's what's driving the reaction? So it's worth pursuing, but it's hard.
And then I also wanted to just finish by asking you each a question about kind of where it becomes personal. So like I know that Mac works on endometrial cancer and telomeres, and I know that you work on breast cancer and other forms of cancer. And so sometimes I'm sure you must have in your lives have had some friend or some relative who says to you, can you read my telomeres? Can you look at some statistical analysis of all my medical data? Can you help me as a kind of one-off? And what do you do when like a person comes to you and says, can you be my diviner, and help me? And so who wants to
go
answer
first?
No, thanks. So, I'll be honest, mostly I ask questions and listen, and make clear that I'm not the person to really...
Right.
I'm not a doctor, and I can't give medical advice. I find exactly these discussions that we're having now about sort of like what's sort of the limits of what we can know.
Right.
Like if I did learn this information, how would it be really useful, and sort of walk through some of those scenarios.
Yeah.
Again, I'm a statistician, so you give me a data set, I'm going to find something fun to do with it.
Right.
So I can build models to predict things, and I'll try it this way and that way, and which one works better. That's all kind of interesting to me because I'm a geek. But really, when I think about like why am I doing this, the first question is, okay, how are people going to use this model?
Right.
So like I can build a breast cancer risk prediction model, but if it's not going to change anybody's behavior, if it's not actually going to help people prevent people dying from breast cancer, maybe I move on and do my day job, spend doing something else.
Okay.
So in the breast cancer example, you can build a reasonable case that, in fact, we can identify women who are particularly high risk, who are walking around not knowing that right now, who would benefit from starting screening earlier or maybe coming back more often. Or for prostate cancer screening, there's tons of side effects that come with screening, right? And overtreatment. So if we can identify a subset of men who are actually at lower risk and can get away without doing PSA screening, that's also a win. But the flip side is, if your intervention is, I'm going to give you a pamphlet on exercising, or I'm going to give you a pamphlet on why smoking is bad for you. It's like we could be giving everybody that pamphlet.
Right. It's bad for everybody. Right. It's just particularly bad for someone else. Yeah.
So I don't know. I think talking about people generally sort of what, just in broad terms, what could you learn, and then what would you do with that information? I think is always interesting.
Interesting, the pamphlet thing about, there are some people, it's not enough to say, I should stop smoking because I know 10 people who smoke and live to their 90s. Mm-hmm. So they still want, some data to say,
for me.
Exactly.
Mm-hmm.
And, but I agree with you on principle. Everyone, you should stop smoking and you should eat better and, go for a walk or something like that. But you will be faced with that person who says, but what, I know 10 people or 9, these crude statistics that they throw out at you. My grandfather lived to be 100 and he ate and drank and smoked anything he could get his hands on. So there's still those people that, and it's interesting, I didn't know this, but what you said about the model for you, it's if there's no, if someone's not going to use it for the purpose that it's intended to, you'll move on to something else.
It's not really going to help anybody.
Yeah, it's interesting because I'm always, the question for me is the how, the why.
Yeah.
I'm okay. So then there's there's many things we need to consider. Then there's, you have a model or a prediction model that could be as great as anything, but you have no intervention or no screening. That is, in my opinion, a sort of a worse situation because now you're telling someone, or you don't tell them, but because you're in your little lab.
Even in physics, right? There's often this problem where people are doing research that is so basic physics that there's like no known application or no known way to observe, 13 dimensions that are hidden from
Right.
view of intergalactic space, et cetera, right?
Right.
Okay. But someday there might be, right? So somebody sort of has to do that. So I could imagine, though, that in the medical world, it's a little bit harder because, no one's life depends on whether or not I can see hidden dimensions in space. But someday it might, the translational work from physics to medicine has a very long history,
Absolutely.
right? So who knows? Maybe that's how we escape the warming Earth. We travel through some hidden dimension to another universe. I don't know. That's kind of crazy, but So anyway, you don't know what the long-term implications of really basic research is.
Right.
Yeah, and to be fair, I have to give a plug to basic research because, as Pete said, a lot of the oncology folks, they're using these sort of tests, genetic tests,
Right.
to target for treatment and even prognostic value. They have prognostic value because if you have this translocation or it's like, ooh, yeah, they get some
Right,
understanding.
and in some cases, I imagine people who particularly are at high risk of dying soon are willing to be part of very experimental studies, and they're sort of. I knew one, unfortunately, friend of mine who died in this way, and he became like a partner with his doctor at Columbia, and
Right.
just do anything you want to me. I'm going to die anyway, and
Right.
just do anything. And so I imagine you can get a lot of data that you wouldn't get, if people wind up in these very odd situations. Which actually brings me to my HIPAA question for you. So if you're, not really HIPAA, but sort of just general ethics, if you're doing a study where you're supposed to be watching, right, but you actually know, like one of your predictions is good enough that you actually know that something bad is going to happen to somebody, you're not really supposed to interfere, right? So what do you do? Because you're not the medical doctors. What happens if in the middle of a study,
Yeah.
and it doesn't even have to be one individual person, let's just say you discover something, but then if you actually go and tell everybody and they change it, it's going to ruin your long-term data and your what do you do?
So, first I would frame it as, if we don't contact somebody, it's not because we don't want to mess up the data.
Right, okay,
This
good.
is not
Good.
we're following people and
Good.
but if we intervene, then we're not going to get crazy.
Okay.
Like we want to see
Good.
the pure natural experiment. That's not why we're doing
Okay.
it. I think there are honest ethical questions and about returning results. This is also a topic of much debate right now
Okay.
with
Yeah.
people on different sides. So people enroll. Maybe when you enrolled them, you had no idea that within 10 years you'd be sequencing their genome,
Right.
and you would know whether they carry the BRCA1 or BRCA2 breast cancer genes,
Mhm.
right? So you never ask them, like, if we find these, do you want to know?
Right.
So they never, like, we would, on
There's
some
an
level you'd be,
unanticipated
Right.
ability to predict
Right.
something. Yeah.
so, maybe they actually don't want to know. And like for some people, ApoE, which predisposes to Alzheimer's or Huntington, I do not want to know.
Because you can't do anything about it right now.
For hunting, right?
Right.
Yeah, right. So if they had been given the choice, they would have said, "Don't tell me. If you
Right.
find out, don't tell me." So we would be presuming in that case by going and contacting them. And the other is, as much as we try to be accurate, research data is not the same as clinical data. Like
Right.
files, like plates get turned, samples get swapped, they get contaminated. So, at a minimum, you would want to send it off. You want to get another sample from that person and send it off to a medical lab to make
Right.
sure that the test was right.
Okay.
Right, in my lab, that's exactly right. We take coverage, so refuge behind the fact that we're a research lab.
Just a scientist.
Just the scientists.
I've heard you say that before.
Yes, we are not allowed to, because we're not a clinical lab. So there is you have to be CLIA certified. There is a procedure and protocol
Right.
to be a lab where you can give out results, and we are not that.
Got it.
So
But that's what I think that model is changing, and one thing that's changing is consents are different now. Like especially in genetics, people
Right.
are being much more upfront about these are the kinds of things that could
Right.
happen, and if we discover this, would you want to know?
Right. And what about, though, these devices? So, in other words, we
Mhm.
talked to Susan Murphy, who,
Yeah.
who
Yeah,
you
yeah.
both know,
Yeah.
uses a huge amount of data from sensors and environmental
Mhm.
data, like literally environmental data, like what's going on around the people. And those people have given consent, but because they really want treatment,
Right.
they're sort of part of that kind of study. But way beyond that, right? Apple right now, it's like, do you want to press that button and let them have your heart rate and your blood
Right.
pressure? And then what happens
Right.
with, people like you in particular, right, can have access to these enormous data sets, but the kind of unanticipated combinations of things can tell you people, to not tell you people, tell you things about people
Right.
that no one imagined when they got the consent.
Right.
Right.
Yeah. No, I mean, because we have explicit consents that we try and walk people through.
Yeah.
We just have real constraints. There's no selling your data to third parties for us.
Hopefully.
No, there's
No.
not.
No,
For
not by
us.
you. I'm talking about the Apples and the
I see.
Facebooks
Yeah.
and the Googles and the whoever and honestly, just I don't know, somewhat off the record. I was on this board on research data and information for the national academies, and we had the ethics and privacy officers
Yeah.
for a number of big companies that I won't name in case we ever do use this. Okay, come and talk to that group. And everyone pretty much agreed that it's the unanticipated combinations of data
Right.
that are incredibly hard to be safe about,
Mhm.
let's put it that way. And so, but the predictability of those combinations is incredible. And so it's really hard to avoid doing that. But anyway.
And back to your initial question, when
Mm.
someone asks me about endometrial cancer,
Yeah,
or
yeah.
masculine, what do I always tell you? I'm not a real doctor.
Yes, right.
And that's how I answer.
Yes, okay, so then we have to get some real doctors in here and ask them the same questions about how they would use
Go
this
on.
information.
Yeah.
So, Nick Patterson, who I'm imagining
Yeah,
you
we
both
know him very well. Yeah.
So we'll talk to Nick again. He and I were Radcliffe Fellows here together, and he is of a very strong opinion that most MDs don't understand uncertainty and that they
Mhm.
don't understand how to use this kind of information. And it's going to be interesting. I think we have to actually talk to some
Yeah.
MDs, and so maybe you can get some of your not too researchy oriented Harvard Medical School friends over here, and we'll talk to them and see how much they value this kind of and how much they understand uncertainty.
Well, because this is my interview and not my
Yeah.
collaborator, there is an oncologist, a pediatric oncologist that I'm working with now,
Yeah.
who's pretty impressive in trying to understand.
Right, so that basically you're working with this person. So basically, we need people who are just treating people
I
who are
see.
very good doctors,
I see.
but who are not involved in research. So I don't want bad doctors. I want
No,
good
no, you
doctors
want,
who are not involved in research and see what they think.
You want like
somebody
a-
you would go to
Right.
if something was wrong with you.
Right, like
but
your
they're not
primary
doing
care.
research. Right. So yeah. Okay. So that was. Oh no, I have one more question. Can I ask you one more question?
Sure.
Okay. So I read this book that is highly controversial because people who know better about this subject or know more than I do tell me I shouldn't read this book. But I like this book, and I've told Mac about this book. It's called Evolving Ourselves.
Oh, yeah.
Have you read this book?
No, I haven't heard
Okay,
of it.
so this book is by Juan Enriquez and Steve Gullins, and what it says is essentially you have your genome, which is immutable over very long periods of time. You need Darwin to change that. Then you have an epigenome, which can change faster, even within your lifetime, right? Then you have a biome, which changes on the scale of days, and maybe even a virome, which could be hours or minutes or something. And that when we talk about the interactions that you were talking about earlier, that there's this kind of internal environment as well as an external environment. And is there any stock in, is this true? In other words, should we actually be modeling changes on a minute to minute, hour to hour time scale in things that are going on that are not the things that are going to change very slowly, but the things that could change more rapidly? Is that part of the future of prediction models, or is this just the opinion of two people who wrote a very interesting book?
There's a lot there.
Which parts of it might be right?
Well, the fact that your germline, your genes, what we consider our genome,
Yeah.
let's go with those that code into a protein, pretty immutable, right?
Yeah.
But then you do have epigenetic areas,
Right.
the telomeres, for instance,
Right.
that are changing, but not very quickly,
Yeah.
right?
But over your lifetime.
Definitely over lifetime, because I'm not going to give you data on telomere, but there's a lot there.
At some level, it comes back to training data, right? Because each of those systems is incredibly high dimensional
And so you need a
and
huge amount
changing
of
over
information.
time. And some of them are changing over the course of a month. Some are changing day to day. Like
Right.
that's why we get fasting bloods as opposed to
Right.
after you've had your had your bagel sandwich for the morning, right? So modeling all that is going to take a lot, and the big question is how much is. I'm not going to say it's not going to. You won't learn anything.
Mm-hmm.
The question is, what do you learn sort of beyond the tools that we have now?
Right.
Like, is something else that you measure now a proxy for
Right.
that, and it's good enough,
Right.
like for your viral
Right.
or something?
You know,
like
and
that.
then I am at the School of Public Health, so wearing my public health hat,
Yeah.
thinking of the things that the major public health challenges that we have now, like the opioid crisis or maternal mortality.
Obesity.
Right. So these are things where you can look at the policy level and the level of the clinic, and you can come up with some interventions that would make a huge difference now.
Right now, right.
Right.
So that other stuff is maybe in the basic research
Right.
for the future category.
Yeah, and
Right.
in terms of, I'm going to use the term somewhat advisedly, wellness.
Okay.
So like living our healthiest lives.
Right, right.
Again, I think there's enough that we know now that's sort of evidence based that
Right.
can help with that. And again, a lot of it is outside you can micromanage your gut microbiome.
Right.
But if you're living in an environment where the roads aren't fixed and the traffic is terrible and there's smog all
You
the
got
time,
bigger
and things-
dangers than
yeah,
that.
Exactly right.
Right. Right.
Yeah.
Right. Right. But your work on the telomeres, that changes. You can see changes year to year.
Not necessarily.
Yeah.
I don't have the data to show you
Right.
can change year to year, at least detectable by
Okay.
the techniques that.
Right. So what's the time scale where it would be detectable?
Well, I could give you data that I often present, and so if you start out between 8 to 10,000 base pairs of these telomeres, there are papers to show that the first 20 years of life you lose about 2.5 kilobases.
Okay.
I mean, and then
Like a quarter.
Okay.
that's on average. This is just
Right. Right.
again.
But then it must be true that in an individual person you do have changes year to
Well,
year.
that's
You
why I
just
say
can't
so. The
easily measure.
genome aspects, the
Right.
genes, which again
Right.
give you proteins and what we consider, they're immutable.
Right.
But the epigenome, this is
Right.
telomeres would fall under that, and
Right.
so yeah, we do see changes. But the year to year, again, as Pete has said, is just so many things you have to
Right,
consider.
but then statistically, then you find these correlations with
We
the
do.
length and the longevity of people. And so somebody might come to you and say, go measure my telomere length
Yeah.
and tell me how long I'm going to live, and you don't want to do that.
No.
Because you don't want to make a prediction based on one measurement for one person and then, right?
No, and I also know
Right.
that it's not just the genes related to telomere, but there are other genes that control other processes that
Right.
also interact with the genes that maintain your telomeres that are
Right,
are
and so the
impacted.
problem
So
is
it's just so
when
too-
somebody comes and they take your research paper and then they make some test where they're going to measure this and then
Right.
they're going to sell it to people and you're going to
people already, they're not using mine, but
Yeah.
there are telomere tests out there, just like there are a lot of other kind of pseudo-diagnostic tests out there. So there's no way around that.
All right, well, we're going to stop now, but if you two want to say anything about these pseudo-diagnostic tests, be my guest.
Well, I mean, so I think it was just last week, 23andMe released a
See, there you got it out of us. I knew it.
a polygenic score for your risk of type 2 diabetes.
Uh-huh.
And so it's actually somewhat controversial. When they like 4 or 5 years ago, they were reporting back these risk scores for a bunch of different diseases, not just type 2 diabetes. The FDA sent them a letter to stop doing that.
Right.
They've now come out with this test, but they're marketing it as a sort of a wellness thing. So which is sort of, and they're not saying don't take any, we're not telling you to take any medical action. We're just saying, you know what, if you're at a little bit higher risk, consider exercising more, or you should lose some weight. So on some level, that's good advice for everybody. Like maintain a healthy weight, eat healthy as much as we know how to do that.
Right.
So the question is, what is this adding?
Right.
And again, how are people going to use this information? We really don't know.
Seems like we need a big educational program.
Yeah. I mean, we will. It's
Maybe
going to be a
something
learning
called
process.
PredictionX.
Right. Yes.
Maybe.
Yeah.
I think you have folks need to have sort of even a rudimentary understanding of risk and statistics.
Right.
And, you know, that can happen, but it will take time. And I think that is necessary to understand something like a 23andMe, because just because you have this doesn't mean, you know, you will get this. And it's
People play fantasy sports. They can understand risk.
That's true. We have to make it. About
it's true.
sports.
And let me up the thread, up the sure. So maybe we can sell it as a fantasy football kind of game, or to make them understand their 23andMe.
Ooh. Okay. On that note, anything else? That
I don't know.
Anything else we here? This was my list of questions. I can reveal it to you now. That was what I wanted to talk about, so you two can look and see if we didn't talk about
Ooh.
something on that list.
I see what you have there. Yeah, I think we covered everything.
Yeah, no,
What are
that's
your
great.
notes?
That was great.
You good?
Actually, yeah, no, we can talk about the association between ApoE and whether you like margarine or butter.
Oh wait, please. We need to hear
Okay.
that. Okay. What is the association that you're talking about?
So, and this sort of gets back to what we were talking about earlier with the training data. Like, where is it coming from? What are the weaknesses in your
Right.
training data set? What's missing, or what's there that really shouldn't be? So there's this fabulous study called the UK Biobank. It's 500,000 people in the UK. They enrolled them. They did really detailed phenotyping. They got detailed body scan measures,
Is this
and they
like
took
for the
their genetics.
NHS?
No, it's its own separate
Separate thing,
Yeah,
okay.
but they link to NHS records, right?
Okay.
And then they link to NHS records, so you can get all kinds of disease
Okay.
information. And they, you know, the people are sitting there, so they gave them a touchscreen questionnaire, and one of the things they ask is like, do you like margarine or butter? Because as one does. You know, so now you can do these genome-wide association studies of all these different traits, including do you like margarine or butter? And sure enough, the ApoE locus, which is associated with cholesterol levels and Alzheimer's disease, is really robustly associated with whether you like butter or margarine.
I like butter.
Yeah. Oh, good. Butter's good. Yeah.
That's butter's good.
Yeah.
Okay.
So now, you know, we think we know kind of sort of why these ApoE alleles are associated with cholesterol levels. Why they're associated with Alzheimer's is still an area of a lot of research.
Okay.
Nobody has ever hypothesized anything about, well, no, of course you're going to like margarine because this is your taste receptor, whatever. So the strong suspicion is that this is a form of bias that's in the data, that people who have high cholesterol
Yeah.
are told by their doctors.
Oh, not to eat butter.
Yes, exactly.
Right,
Stop
to
with
eat
the butter
margarine.
already. Eat margarine.
Yeah, but
But setting aside whether that's good advice or not,
Right,
but that's the
And
advice
then
they were
decide
given.
they like
so
it.
So then that's why people are. So you give high cholesterol, and that's what's driving to whether
I'm
you
going to
like
stick to
margarine or
physics,
butter.
okay?
Yeah, right. Observational data is fun. Yeah.
right. But see, that will make headline news, and every phone will light up with every notification.
Yeah.
And then the poor people who study nutrition are like, oh God, we got another one to deal with, because that's newsworthy.
Right. So, I mean, I guess, and part of the point of that example is the association between ApoE alleles and whether you eat margarine or butter. It's real. It's there in the population.
Right.
But the explanation is sociological.
Right, exactly. In a different
Exactly.
context,
Right,
in a world where nobody's giving you advice about whether you should eat butter or
Probably
margarine, there's no
correlation.
association.
Right,
Right.
yeah.